Hello dear students, welcome back to Biovidyalaya. I hope you all are doing very well. Today we are going to discuss about cell adhesion molecules. So what is meant by cell adhesion? Cell adhesion means it is the binding of a cell to a surface like extracellular matrix or any other cells. How these cells are binding to this extracellular matrix or any other cells? Yes, these cells are binding to extracellular matrix or to any other cells using some molecules or proteins known as cell adhesion molecules, or we can call it as CAMs. So, cell adhesion is the binding of a cell to a surface, extracellular matrix or another cell using cell surface proteins called cell adhesion molecules, CAMs. So, cell adhesion is the binding of cell to extracellular matrix. Extracellular matrix means it is the network of proteins and other molecules that is surrounding the cell and that is giving support and structure for the cell. Okay, that is called extracellular matrix. So cell adhesion is the binding of cell to extracellular matrix or other cell using some proteins called a cell adhesion molecule or CAM. These CAMs can be cell-cell adhesion molecule or cell matrix adhesion molecules. Okay. So these CAMs are typically transmembrane receptors and are composed of three domains. CAMs are transmembrane proteins and they are composed of three domains: intracellular domain, transmembrane domain and extracellular domain. See here, this is extracellular domain, this is CAM and this is the extracellular domain and this is transmembrane domain that is passing through the plasma membrane, and this is intracellular domain, that means cytoplasmic domain. Okay, so this intracellular domain that interact with the cytoskeleton, that means that is interacting with the protein filaments present in the cytoplasm like actin or intermediate filament. Okay, and then transmembrane domain that is passing through the plasma membrane, and the extracellular domain that is interacting with CAMs of the same kind or with other CAMs of different kind or with the extracellular matrix. Okay. So here you can see that this is CAM and this is also CAM. This is I will write here. This is the CAM present in cell 1 and this is the CAM that is present in cell 2. Okay. So these are the same kind of CAMs, isn't it? So this interaction is called a homophilic interaction or homophilic binding. Okay, because here the cell adhesion molecule between two neighboring cells are identical. So it is called a homophilic binding or homophilic interaction. Here you can see that this is one kind of CAM and this is another kind of CAM, isn't it? Okay. So here CAMs are different kinds of CAMs are interacting. So it is called a heterophilic interaction. This is homophilic and this is heterophilic. Here cell adhesion molecule between two neighboring cells are not identical. That's why it is calling heterophilic interaction. Okay. So extracellular domain that interact with other CAMs of the same kind or with CAMs of the different kind or with extracellular matrix that will also interact with extracellular matrix protein. Okay. And CAMs are classified into four major families: integrins, immunoglobulin superfamily, cadherins and selectins. Okay, this you have to remember. There are four major families of CAMs: integrin, immunoglobulin superfamily, cadherins and selectins. Okay, so let's see about cadherin. Cadherins are calcium-dependent cell-cell adhesion molecule that mediate adhesion between cells and tissues in organisms. So, cadherins are calcium dependent that need calcium for their activity, and they are cell-cell adhesion molecule that is helping cells to bind each other. Okay, so cadherins are calcium dependent cell-cell adhesion molecule that mediate adhesion between cells and tissues. Okay, Cadherins have extracellular cadherin domains, that means easy domains in the extracellular region. There are calcium binding sites between each pair of easy domains. Okay, see this picture. Here you can see that this is cadherin. Okay, here you can see that this is C-terminal end. This is C-terminal end. This is transmembrane domain. This is transmembrane domain and this is extracellular domain. Okay. And this is called the easy domain. These extracellular cadherin domains are called the easy domain. This is EC1. This is EC2. This is EC3. This is EC4 and this is EC5. These are easy domains. In between these easy domains, we can see calcium binding site. This is calcium binding site. Here back calcium binding site is present. Here, here and here. So in between these EC domains, calcium binding site is present. So cadherins have extracellular cadherin domains called EC domains in the extracellular region. This is extracellular region and this is intracellular region. There are calcium binding sites between each pair of EC domains. Okay. N-terminal tip of one cadherin molecule bind the N-terminal domain from a cadherin molecule on another cell. So this is the N-terminal end. This is the N-terminal end and this is C-terminal. Here also this is N-terminal and this is C-terminal. Okay. So N-terminal domain, that means extracellular domain of one cadherin that will bind with extracellular domain of another cadherin on another cell. Okay, and the C-terminal tip interact with the cytoskeleton via adapter proteins like beta-catenin, alpha-catenin, vinculin and plakoglobin. Okay, so this is a C-terminal end. This is the C-terminal end. So this C-terminal end will interact with cytoskeleton. See here, this intermediate filament and actin filaments are um, cytoplasmic fiber. They are cytoskeleton. Eh? Okay, so this C-terminal end of this cadherin molecule will interact with cytoskeleton via adapter protein. This is adapter. This is also adapter protein. Example for adapter protein are beta-catenin, alpha-catenin, vinculin and plakoglobin. Okay, so this extracellular domain or easy domain that will interact with easy domain of another cadherin on another cell, and the C-terminal tip will interact with cytoskeleton via adapter protein. And these cadherins are showing homophilic and heterophilic interaction and heterophilic. So these cadherins are showing both homophilic and heterophilic interaction. Okay. That means cadherin can interact with other cadherin molecule or they can interact with any other Ca molecule. Okay. And these cadherins are differentiated into several types based on their location and properties. That means classical cadherin and non-classical cadherin. Classical cadherins are two types, type 1 and type 2 cadherins. And these type 1 and type 2 cadherins are named on the basis of tissue within which they were identified first. First is type 1 cadherin. Example E cadherin, N cadherin and R cadherin. E cadherin means epithelial cadherin. E cadherin means epithelial cadherin. That means they found in epithelial tissue. N cadherin means neural cadherin. Neural cadherin that is found in neurons, and R cadherin means retinal cadherin. Okay. Next is type 2 cadherin. Example VE cadherin. VE cadherin means vascular endothelial cadherin. Vascular endothelial cadherin. Okay. Next is non-classical cadherins. First is desmosomal cadherins. Desmocolin and desmoglin. Okay. So desmoglin and desmocolin are desmosomal cadherin. They are forming cellular junction called the desmosome. They are forming cellular junction called the desmosome. Okay. And this easy domain of this desmocolin and desmoglin that will responsible for adhesion, and the intracellular region, that means the C-terminal end will interact with intermediate filaments via adapter protein called the placoglobin and the desmoplankin. Here you can see that this is desmosomal cadherin, this is N-terminal end and this is C-terminal end. This C-terminal end will interact with intermediate with the help of some adapter protein like placoglobin and desmoplankin. Okay. So this desmocolin and desmoglins are forming cellular junctions called the desmosome. Okay. And next is atypical cadherin. T cadherin and LA cadherin. T cadherin means that is also known as H cadherin or Cadherin 13, CDH13, Cadherin 13. And this T cadherin that lack transmembrane and cytoplasmic domains, and it is anchored in cell membrane through the GPI anchor. Okay. So this T cadherin that is lacking transmembrane and cytoplasmic domain, and it is anchored to cell membrane through GPI anchor. Okay. And LI cadherin means liver intestine cadherin or CDH17. That means cadherin 17. Okay. And this overexpression of this CDH17 can be detected in cytoplasm of liver cancer, gastric cancer and colon cancer cell. Okay. And next is protocadherin. Example PCDH alpha, PCDH beta and PCDH gamma. These protocadherin are most commonly found in the nervous system, and their diversity may contribute to nervous system development and organization. And these proto-Cadherins are showing homophilic cell interaction. Homophilic cell interaction. Okay. Next is immunoglobulin superfamily CAM or IgSF CAM. IgSF CAM, immunoglobulin-like cell adhesion molecule that mediate calcium independent cell-cell adhesion. That means that does not need calcium. This family is characterized by their extracellular domains containing Ig-like domains responsible for adhesion. Here you can see that this is IgSF CAM, and here in their extracellular domain you can see immunoglobulin-like domain, and this immunoglobulin-like domain only interacting with this integrin. So these immunoglobulin-like domains are responsible for cell adhesion. Okay. And here also you can see that this is NCAM, and here you can see that immunoglobulin-like domain, and this is fibronectin type 3 domain. Okay. And this immunoglobulin-like domain only interacting with another CAM. Okay. So this family is characterized by their extracellular domains containing Ig-like domain responsible for adhesion. The members undergo both homophilic and heterophilic adhesion, has the ability to bind integrin or different IgSF CAM. Here you can see that this IgSF CAM is interacting with integrin. Okay. So this is called a heterophilic adhesion. Here IgSF CAM is interacting with another IgSF CAM. So it is called a homophilic adhesion. So the members undergo both homophilic and heterophilic adhesion. Example for IgSF CAM, ICAM, VCAM and NCAM. ICAM means intracellular cell adhesion molecule. VCAM means vascular cell adhesion molecule, and NCAM is neural cell adhesion molecule. And ICAM and VCAM on endothelial cell mediate heterophilic binding to integrin on WBC. We know that during inflammation, ICAM is expressed on endothelial cell. ICAM and VCAM are expressed on endothelial cells that will bind with integrin on leukocyte, and that is facilitating the transendothelial migration of leukocyte to the inflammation site. Here you can see that here VCAM is there, that is binding with integrin that is present on leukocyte. Okay, that will helpful for transendothelial migration of leukocyte to the inflammation site. Okay. Neural cell adhesion molecule NCAM, also called CD56, is showing homophilic binding. That means this NCAM will bind with another NCAM on another cell, and it is expressed on the surface of neurons, glia and skeletal muscle. Okay. So here ICAM and VCAM on endothelial cell mediate heterophilic binding to the integrins on WBC, and NCAM that is showing homophilic binding, and that is expressed on the surface of neurons, glia and skeletal muscle. Okay. Next is selectin. Selectins mediate cell-cell adhesion by heterophilic binding. That means selectins will bind with another CAM molecule. Okay. That is called heterophilic binding. And selectins are calcium dependent, that need calcium for adhesion, and they are single chain transmembrane glycoprotein. Okay, here you can see that these are selectins. Here this is N terminal end and this is C terminal end. In this N terminal end you can see lectin domain. This green color one is lectin domain, and that is followed by EGF. That means epidermal growth factor. Epidermal growth factor domain. Okay. So this is epidermal growth factor like domain, EGF domain, this orange color one, that is followed by consensus repeat unit. Okay, and this is transmembrane domain, and this is C-terminal end, cytoplasmic tail. Okay. So they have conserved lectin domain that binds to a specific sugar polymers on another cell. That means this lectin domain, it will bind with specific oligosaccharide that is present in another cell. And so these lectin domains are responsible for adhesion. Okay. Three non-members of selectins are L-selectin, P-selectin and E-selectin. L-selectin present on white blood cells, P-selectin present on blood platelets and endothelial cells, and E-selectin present on endothelial cells. Okay. Here you can see that this is L-selectin. Okay. And here you can see that 1, 2. Two consensus repeats are present in L-selectin. Here this is E-selectin. Here how many consensus repeats are present? 1, 2, 3, 4, 5, 6. 6. Okay. Here 2. Here 6. Okay. And here this is P-selectin. Okay. This is P-selectin. Here 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 consensus repeats are present. Okay. Last one is integrin. Integrins are transmembrane glycoprotein, and integrins are the principal receptors used by animal cells to bind to the extracellular matrix. That means that is helping in cell matrix adhesion. Okay. They are heterodimers composed of alpha and beta subunit and function as transmembrane linkers between the extracellular matrix and the actin cytoskeleton. Here you can see that this is dimer, that is heterodimer. Okay, alpha, and it is composed of alpha and beta subunit, and here it is interacting with extracellular matrix protein like collagen, fibrinogen, fibronectin, etc. And here this is C-terminal end. This C-terminal end is interacting with actin filament. Okay. Cytoplasmic portion of each integrin molecule bind to adapter protein like taline, alpha-actinin, vinculin that interact with the actin filaments inside the cells. Here you can see that this C-terminal end of this integrin, that means cytoplasmic portion of integrin molecule is binding with adapter protein like alpha-actinin, taline and vinculin. That is how it is interacting with actin filament. Okay. And this extracellular portion, that means N-terminal end of the integrin, that is binding with fibronectin, fibrinogen, collagen in the extracellular matrix. And the binding of integrin to their ligands depend on extracellular divalent calcium, Ca2+ and Mg2+. Okay. And integrin also serves as cell-cell adhesion molecule, helping the cells bind to other cells. Okay. Here you can see that during inflammation, ICAM is expressed on endothelial cells. Here you can see that ICAM. This is ICAM. I will show you. Here You can see that ICAM is there. Okay. And this ICAM is binding with integrin on leukocyte. Okay. This is integrin and it is present on leukocyte. This is leukocyte. Okay. And this binding is helpful for the migration of leukocyte to the inflammation site. Okay. So ICAM expressed on endothelial cells is binding with integrin on leukocyte and that is helping in the migration of leukocyte to the inflammation site. So integrin is helping in cell matrix adhesion and also cell-cell adhesion. Okay. If you like this video and find it helpful, then please click the like button, share this video with your friends and also don't forget to subscribe this channel for getting more videos like this. Keep learning. Thank you.